And so with that, I am going to move on to the functionality. I really want to highlight about MEBS and the things that you can do with MEBS. Here are some of the examples of the things you can do with the entropy scores. You can map those entropy scores across the tree of life or phylogenetic trees. You can cluster those scores and have similar related genomes. You can map those scores across time, and as I was mentioning with the sulfur score, you can map them if you have a geographical localization of that. But let's talk about the PFAM entropies, and I'm glad that the question is related to this question. So here I have protein families, and this is one of the examples of how useful the relative entropy can be. Here we have the entropies computed across 2,000 genomes. And here we have the distribution of the entropies if you calculate these entropies randomly, if you don't consider organisms that are enriched in those cycles. So if you compute the relative entropies randomly, this is the distribution that you get. And then you see, and this is the entropy mean, then you see that there's some deviation of the entropies that are informative for that. They are let's say separated from the random distribution. The good thing about this is that we, by using this analysis, we can get or capture proteins that are domains of unknown function that are very enriched, or let's say conserved in organisms. In this case, are for nitrogen. Yes, this is for nitrogen. And so you can see just without knowing anything, you can get the entropy values and see what are those proteins. We have other proteins that we know that they have been characterized and they are involved in the nitrogen cycle. But if you use this approach, you can get proteins of domains of unknown function. And this is another representation of that. Here is the distribution. If you calculate the relative entropy randomly for each of these PFAMs thousands of times, and then we see here a random, the distribution that it's away from that random distribution, sorry. The values that are far from the random distribution. And this is implemented in MEBS. These scripts are scripts that you can use already, and they are under the scripts directory, and that you can easily get these plots if you use MEBS. The other thing, and I'm going to give you real life examples of the scores. For example, here our postdoc Dr. Gong is analyzing novel bacteria from different environments around the world, hydrothermal vents, cold seeps, and coastal sediments. And we don't know anything about this bacteria, but just by mapping the scores across this bacteria, we can see which ones can be enriched in, for example, iron cycle. Here, iron cycle, this don't seem to be very enriched compared to the rest, but here we can identify specific organisms that can potentially have some implication in these cycles. If they have a negative or a very low score in oxygen, we can have some insights about the physiology of this or the metabolism of this bacteria that are not oxygenic. Maybe these are anaerobic bacteria. So you can get an insight just by mapping the scores across phylogenetic related bacteria. And this is one example that we've been working on. And so this idea about discovering novel metabolisms by using the scores is really powerful, because MEBS we have already pre-computed the scores of a data set of 2,000 genomes, and we have the values of those. So if you use that information that it's already in MEBS, And if you use this script that is in MEBS, you can use this script with any type of data you have. And it's really powerful because it allows you to visualize your data set in 3 different ways with different projection methods. We have isomap. Here I have the projection of isomap. Here we have TSNA. And here we have a PCA, which is the most commonly used. And here we have the 4 clustering methods. And so just by using this script, fMEBS cluster, with a data set, with any type of data frame, with columns and values, you can have a clustering of your data and visualize your data. So now imagine that I want to focus specifically on this type of data, on this graph, because this graph, let's say, separate the clusters the way I want it, right? And so with the same script, we can have that specific graph, and we can get the genomes that belong to each of the clusters here. And when we analyze this, when we analyze the organisms that belong to these clusters, we can see that we can have some sort of metabolic relateness just by plotting the scores. And the scores are values that inform you about nitrogen, oxygen, iron, sulfur, and methane cycle. And another way to represent this is by using the other script that it's meant to visualize your data, but in a reduced way. It just take into account the standard deviation of the data. And it's basically the same projection, but kind of reducing that data set in another way. And this was very useful for me because when I started my postdoc, we had information about this archaea that were very unique, and they were not similar to anything that we have seen before. And this kind of pre-analysis allowed me to see that, for example, my organisms that are Brocaria were not related to methanogenic archaea. They were not even similar. They were related to archaea and anaerobic archaea and fermentative bacteria, but they were very far from methanogenic archaea. And this was very important for me to understand at first this very unique organisms that are able to metabolize methylated compounds, as we can see here. The methylated compounds are very abundant in the atmosphere, in the oceans, but in archaea, until now, we thought that only methanogenic archaea was able to use methylated compounds. Nothing outside the methanogenic world was described to use methylated compounds. But we knew that they were not methanogenic, and also because they didn't have any of the genes, nothing related to methanogenesis. And so that was very important. And we also used MEBS in order to map all of the genes that were important for these organisms, the ones that we were interested in, and the ones that we wanted to focus. And another example of how we use MEBS, and this is a paper that is really, really pretty. And this was Maggie's master thesis, and she's amazing. She did an amazing job with this paper. We basically put the MEBS scores of groups of organisms that were similar metabolically. And as you can see, for example, here are the oxygen scores, the sulfur scores, the iron scores, the nitrogen scores. So we can see, for example, this was very important for us to understand one group of bacteria that was very important for iron cycle. And then when we look at the genes that were belonging to those group of genomes, we indeed confirm this. So this massive analysis and comparing thousands of genomes And just by plotting the distribution of the score is really powerful to then zoom into the specific data. Because sometimes when you're analyzing metabolism, it's overwhelming and you don't know where to start. And this is a way you can start. Also, the other information you can get with MEBS is the pathway completeness. And the pathway completeness is basically, it's going to give you how complete are several pathways that are previously pre-computed in MEBS. This is something that you can get if you use MEBS with your genomes here. Ian is analyzing a time series analysis with hundreds and hundreds of organisms, archaea and bacteria. And with this graph, we were able to kind of separate them and focus on the ones that are important, for example, for ammonia oxidation, and the ones that have specific pathways, and all of this is based on the PFAMs. And this information you can use to plot those values into the tree of life, and also have well-defined groups and see whether there's a phylogenetic signal or a metabolic signal just by putting that data into your tree. And another functionality is also that the pathways that you are analyzing, you can screen those pathways. Imagine, in this case, I separate all the pathways involved in the sulfur cycle, and I screen all those pathways across thousands of genomes, 2,000 genomes, and 900 metagenomes. And then I want to see how complete they were. And then I wanted to focus, and so the first thing you notice is the pathway completeness. Obviously, it's more going to the right side is more complete in metagenomes because we're analyzing communities, and obviously communities and organisms are going to have most of the genes involved in those pathways. And compared to the genomes, right? But there are genomes that have most of the pathways, most of the genes for those pathways. And this is a way in which you can screen several pathways at the same time, plot the results, and then focus on the ones that you're interested in. If you want to focus on the genomes that have the most complete pathway for, I don't know, sulfate reduction, or pathways involved in nitrogen fixation, this is a way that you can do it. And finally, not finally, there's two more. Another thing you can do with MEBS is genome clustering. And this is a function that we just implemented for Maggie's paper. Here I am showing you a phylogenetic tree that is in preparation. Also, Dr. Shen Gong is working on this. And here we have the different scores across this bacteria tree. And then we have the cluster groups. And here, here are the MEBS clusters. So these are groups of genomes that are metabolically related. And how do we do that? We basically cluster the genomes based on their protein content, meaning for each genome, we screen all of the protein families. Right now, the PFAM database and the one that is incorporated in MEBS, it is the PFAM version 34 that has more than 19,000 PFAMs. So imagine that you have a presence-absence matrix of 19,000 PFAMs and the presence-absence in all of those genomes, and basically using this script, this algorithm, to cluster them and to kind of collapse the genomes based on their metabolic relatedness. And then you can plot those groups. For some cases, as you can see, these groups, there's a very clear phylogenetic signal of metabolically related groups, but sometimes it's not that clear as we can see. Oh my God. As we can see here, sorry, in the orange and in yellow. And the same thing, that's what we applied for Maggie's paper, for the Delta paper. We analyzed almost 2,000 genomes, Delta Proteobacteria and non-Delta Proteobacteria. And we pretty much did that. We analyzed whether there was this phylogenetic signal. Here in this group in yellow was the one that have the very high iron scores, and they are very restricted to a specific group in the phylogeny. And then we have other groups that are spread throughout the phylogeny. But here we can have an idea of how metabolically related are those genomes with PFAM clustering. And lastly, the last thing that it's incorporated in MEBS is that you can evaluate and identify your the bins that you have metagenome assembled genomes based on their completeness of a specific single marker gene. So one of the questions that you ask once you have your genomes, your bins derived from your metagenome is how complete my genome is, how much of the, what fraction of this genome was captured, what is the taxonomic identity of these organisms, how complete is that genome? And for that, there are several programs that allow you to do that. One of them, the most used, is CheckM. The other one is MyComplete. And so they do that. You put your genomes, either FNA or nucleotides or amino acids, and then you get a value of how complete your genome is based on this specific set of marker genes. And there are a set of genes that are suitable, or they are good for conducting this kind of analysis to evaluate completeness. They have to be present in all the organisms in single copy. Not every time is the same, as we are now discovering. And they don't have to be universally horizontally transferred, right? Because they have to reflect the evolutionary signal of an organism. So, yeah. So now what I did was to compare. I compared the values that you can get with MEBS. So I incorporated into MEBS a set of a specific single copy marker genes from archaea, from bacteria, from both. And I incorporated the marker genes that are described in MyComplete. And I put that into MEBS, and I calculate the completeness, and I compare the values that you can get the completeness with those other two programs. And as you can see here, for each bin, we have all these different values. And as you can see in this graph, the values are very similar. So this means that with MEBS, you can get a similar result as you can get with CheckM or with MyComplete. Here, for example, are the values of completeness for archaea. And this is important because here we know that we are analyzing bacteria, but sometimes you don't know. Sometimes you have bins and you want to benchmark them and you want to know what they are. And so with MEBS, you can have an idea if they have low scores, low values of completeness of single copy marker genes in archaea, then you can screen that and look at the bacteria completeness values. And that's something that, for example, you don't have with the other two programs. So incorporating both genomic completeness and metabolism makes it very powerful in a way in order to make high throughput and large scale analysis when you are kind of starting analyzing your data. And I think with that, I cover all the functionality and steps for MEBS.